Okay, let's get the show on the road and see how this works. And what we're going to try to do is videotape most of these PowerPoints for you and place them to a place where you could watch them and observe them. And hopefully that will help and we'll be able to answer questions. And our topic today that we're going to talk about is basically muscle tissue structure and function. We'll be finishing off with another presentation just following this that will be about how the muscle actually works. So let's go from here, talk about that. Well, first of all, when we look at muscle tissue comprises a large part of my body mass and my body weight. About 50% of my body weight is just from muscle itself, okay? So it's a significant part of our anatomy. The word myology basically has to do with the study of muscles. So when you ever see something that says myology, that's what that means. Sarco is just a muscle prefix, as if you had medical terminology. This is old hat for you, but sarco is just a muscle prefix that means muscle or skeletal muscle. The sarcolemma is basically the plasma membrane or the cell membrane of a muscle itself. I'll be talking about the sarcolemma a lot more in our next lecture on video. Sarcoplasm is the same thing as the cytoplasm of a muscle cell. So where in a regular cell we talked about the cytoplasm, in a muscle cell we call it the sarcoplasm. The sarcoplasmic reticulum, well, guess what? No joke here. This is the endoplasmic reticulum of a muscle cell, and we've talked about the purpose of the endoplasmic reticulum in our first section in this course already. Sarcomere is just another name for the muscle cell itself. Has a very complex structure, which we'll be talking about in our subsequent videos, but the sarcomere is basically the repeating structural units of a muscle cell that are reported and required for contraction. They're very cylindrical appearing. And just another word would be sarcoma. Sarcoma just is a certain type of muscle cancer that we will see. So anyway, this is just a little about general ideas about the muscle tissue. We know that we have three types of muscle. We talked about this in the lab earlier in the year. Skeletal muscle. Skeletal muscle we know is voluntary, which means that it requires an active command from my brain that I actually I recruit this muscle to do something for me. So it's voluntary. It's also striated. When we looked at the skeletal muscle underneath the microscope, we saw it had multiple cross stripes or striations across it, which will make sense a little bit more when we talk about the muscle structure, how the cells actually structured in our next video. We know that what happens is there's so much going on in a muscle. Sometimes it needs a couple of command centers. So therefore, skeletal muscle cells are frequently multinucleate. They'll have multiple nuclei within a single sarcomere. We know they have to be highly vascular. Why? Simply because they're required to, when they contract, it's required to perform work. Work requires energy. Energy requires both fuel as well as oxygen for the energy to be produced. So we know that the skeletal muscle needs to be highly vascular. We know that what happens is when the skeletal muscle will connect one bone to another bone, and it connects to the bone by what we call tendons. We'll talk about that later on. And basically, when the muscle contracts, it allows the angle between two adjacent bones to change. So therefore we know skeletal muscle is used for motion, for posture, as well as for performing various types of work. Cardiac muscle had some similarities but a lot of differences from skeletal muscle. Cardiac muscle is involuntary. There are certain things I could do to actually control my heart rate a little bit. If I hold my breath, because of stimulation of the 10th cranial nerve or the vagus nerve, will actually slow my heart rate down. But basically my heart, for the most part, is working on its own. So therefore it's involuntary. It beats at an inherent rhythm based upon the leakiness of some of the cells in a specialized area of the heart, which we'll talk about when we talk about the cardiac section. We know there's also striated, so it looks underneath the microscope somewhat similar to skeletal muscle because it has those cross striations very much like we saw in skeletal muscle. Cardiac muscle, however, is uninucleated. It only has one nucleus. We know that it's highly vascular. Why? Because skeletal muscle is always under a little bit of contraction, which is what we call tone, but cardiac muscle is constantly working, whether I'm awake or whether I'm asleep. It's constantly moving. So therefore, it always needs a fuel and oxygen supply. So therefore, to deliver that fuel and oxygen supply, we need to have the cardiac muscle being highly vascular. One of the things that we did see when we looked at under the microscope at cardiac muscle was that the muscle fibers seem to sort of branch off, and this is what we see in cardiac muscle. Therefore, one muscle cell will actually branch off that will be able to make contact with multiple other adjacent muscle cells, and where they come together, where the end of one muscle cell branch comes to meet another end of a muscle cell branch from a cardiac muscle, another cardiac muscle. There's a little area that we saw in the microscope, and these are called intercalated discs. There's a little darker areas which go across that muscle cell itself, and we know cardiac muscle means heart muscle. The third type of muscle we have is visceral or smooth muscle, and basically these are involuntary as well. We don't really control these as well. They're done part of our nervous system, which we'll talk about in the neurological section, called the autonomic nervous system, will control these. We'll actually make them smaller or larger. We'll see these in blood vessels. When a blood vessel contracts, it gets smaller, okay, and when it dilates, it gets larger. Same thing happens in the smaller airways that are lined with these smooth muscles in my lungs. So we'll open up bronchi, or these bronchioles, make them all open, or bronchodilation, or we'll make them constrict. We see the people with asthma, one of the problems is these involuntary smooth muscles in the lung become a little hyperactive, and they will start to contract, which decreases the airway into the small air sacs called alveoli in the lungs, which is one of the problems that people with asthma might have. Therefore, what do they take? They take a bronchodilator, which relaxes these smooth muscles, which allow these little bronchioles to start to widen up and get larger to allow more air to get down those air sacs. We know that smooth muscle is non-striated. It doesn't have stripes across it. So therefore, when we looked at it in the microscope, it was basically looked a lot similar to a lot of other cells. It's uninucleate. It's more spindle shaped. It sort of looks like more of a spindle. It's wide in the middle and very narrow and pointed at the ends. And again, we know the smooth muscle is for viscera and blood vessels. Viscera also, we will see where smooth muscle is very important is in the GI tract. When you're sitting there trying to be quiet and your stomach starts to gurgle, basically a lot of that is simply because what's happening is these smooth muscles are contracting to push things throughout and try to get things down to the end of the line. So as a result, it moves things around to the GI tract as a lot. Okay, so we'll be talking a little bit more about smooth muscles later on. So this is a chart we saw before, talking and looking at the major differences between skeletal, cardiac, and smooth muscle. We had this in the lab when we talked about the different types of muscle. So I really don't want to spend much time on this. You could actually look it over, pretty much sort of like a summary of everything we've talked about in this previous slide. So that's what that goes on. Let's talk a little bit about the functions of skeletal muscle. Well, first of all, for me to do something, muscle contracts. So therefore, we know that the skeletal muscles is quite important for motion and for movement, and this is what we call work, okay, and provide some type of a force. So we use skeletal muscle for motion and movement, and therefore this is what we call from a physical nature, both force and work. We also use skeletal muscle for heat production. When you're cold, one of the things you know is you will shiver. And the reason why you're shivering is causing these skeletal muscles to rapidly contract. And what that does is it generates heat. So skeletal muscle is also very responsible in heat production. Maintains posture. In other words, like I mentioned before, all the muscles are a little bit of what we call tone, which means the muscles are mildly contracted at all time. They're not totally flaccid. There's a little bit of a tone in all these muscles. However, to be able to maintain the posture, this tone in certain muscles is very important to be able to maintain posture. Also, in regulating organ volume, skeletal muscle becomes important. Now, this is going to be more important when we talk about how we actually breathe in the respiratory section. And between all the ribs, we talked about that intercostal space when we looked at the ribs in the lab on the skeleton, and the intercostal space is filled with some muscles called intercostal muscles. And what happens is during breathing, these intercostal, particularly the external intercostals, the ones on the outside, will contract, and what that does is the rib above lifts the rib below up towards it, and that expands the chest in both from front to back and side to side, and that increases the volume inside the thoracic cavity, which actually creates almost like a suction or a vacuum, which then draws air in. That works along with the diaphragm that we'll talk about later on. So these are some of the basic structures or the basic functions that we see of skeletal muscle. Muscle characteristics. What does muscle do? First of all, it contracts. So the ability of muscle to be able to contract basically is what muscle's all about, for the most part. It shortens to be able to produce force. In other words, the muscle cell will be a certain length, but when certain things happen within that muscle cell, which we'll talk about in our subsequent video, it actually causes that muscle cell to shorten or to decrease in the overall length. And by doing that, it produces a certain level of force. So the contractility is exceptionally important in regards to a muscle. We know that muscle is excitable. Something needs to tell the muscle to be able to contract, and therefore that's a signal from the brain. Signals from the brain, which we'll talk about in our subsequent video, is transmitted to the muscle surface, the sarcolemma, and actually causes a chain reaction, which then allows the muscle to contract. So it has to be excitable. It's conductivity. The muscle, the sarcolemma, or that membrane, that muscle cell membrane, is able to conduct a certain electrical impulse along it, which allows that muscle to contract. And these are what we call action potential. And action potential is created, which is an electrical signal that actually will cause that muscle to contract. Muscle is also extensible. In other words, we could stretch a muscle without it actually coming apart to a certain point. I mean, this is not infinite where you can stretch it, and because eventually it's going to pull apart. But muscle can stretch to a certain point without damage. And this is important because let's look at the thigh. If I try to extend my knee, just take your foot, take your leg, and if you extend your leg, the muscles on the front or the quadriceps of the thigh are tightening. What happens is the muscles on the back or the hamstrings have to stretch a little bit to be able to sort of coincide with that muscle on the front, the anterior muscles, the quadriceps, contracting or getting shorter. So the muscle that's doing the work, by the way, is called the agonist, and the other muscle that's beyond this on the opposite side is called the antagonist. So as an agonist muscle will contract and get shorter, what happens is the antagonist muscle on the opposite side has to relax and stretch and to get longer. Okay, so that elasticity is really important. And then what happens is because the muscle is wrapped in what we call this fascia, or this elastic band that's around it, it's able to return to its normal shape and form once that stretch is reduced. So these are some of the muscle characteristics I think are important to understand. A little bit about the skeletal muscle anatomy. Grossly, when I look at a muscle, has the biggest portion in the middle is called the muscle belly. Okay, and this muscle belly is actually a series of different bundles of muscle fibers itself, and we have sort of like a hierarchy of bundles, and going to various a larger portion of them, and the smaller down to that. And when we look at this sub-level of bundles, what happens is these bundles are surrounded by again this material we call fascia. Next time you have a roast or something like that you get, or a steak that you're getting ready to put on the grill or in the oven, look at the muscle. The muscle is going to be that reddish tissue, but between the muscle, okay, is this whitish band of material. That's basically fascia, and that fascia sort of holds things together so it doesn't stretch out. So the muscle fiber is contained within this what's called a fascial bag. If I look at the whole muscle itself is surrounded by what we call the epimysium. Okay, epimysium, which is basically surrounds the whole muscle belly itself. What happens is inside that muscle belly are smaller bundles of multiple fibers, and this is the perimysium, which basically has smaller bundles of fibers inside, and then each muscle cell itself is surrounded by the endomysium, which is basically individual muscle cells are surrounded by fascia as well. So the whole muscle is surrounded by the epimysium, which is basically surrounds the whole muscle. The smaller bundles of muscle fibers is the perimysium, which contains smaller bundles, and then the muscle itself, the muscle cell itself, the individual fiber, is called the endomysium. Okay, we talked a little bit before about how the muscle attaches to bone, and what muscle attaches to bone with is by tendon. Tendon's a dense connective tissue, which is really not very stretchable, very only mildly stretchable, a lot less than what we'd see in muscle fiber itself. So the tendons are what actually connect muscle to bone. Okay, we also what's called an aponeurosis. An aponeurosis is sort of like an elongated tendon, which is basically very fascial, much more of a band or a sheet of connective tissue. If you look down the lateral side of the leg, well, you can't see it, you can't really look and see it, but down the lateral side of the band, there's called the tensor fasciae lata, which is largely a large band of fascial material that goes down the lateral aspect of the thigh, from all the way from the hip all the way down to the knee. And this is called an aponeurosis. When we talked about in the skull, there's a muscle in the front which is called the frontalis, which raises my eyebrows and stuff like that. In the back of the skull, there's also a muscle called the occipitalis, and the two are connected over the top of the cranium by what we call the epicranial aponeurosis. Okay. We also know that what happens is muscles themselves are divided into sections. If I look at like the hamstrings, remember we talked about that linea aspera that went down the back of the femur before. That linea aspera is actually a band of deep fascia which divides the hamstrings on the medial side of the thigh, which are two, from the hamstring on the lateral side of the thigh posteriorly, which is one. So the deep fascia allows separation of muscles. Also, because it allows a separation, there's a fat that's around there, and this fat that's around the aponeurosis will allow a path for blood vessels, nerves, and stuff like that to be able to move, to be able to innervate, or to be able to supply the muscle itself. Okay. We also know that deep fascia is sometimes a point of attachment for some muscles themselves. Okay. So basically, this deep fascia will divide muscles, separates muscles, one muscle from another. This so allows one muscle to move where another muscle that might be close to it might not move, and also provides a point of a path for various other structures like nerves and arteries and lymphatics to be able to move through an extremity or part of the body, so it doesn't have to pass all the way through a muscle itself to the whole distance. And this is what we're talking about when we start to look at this. Let me see if I could annotate this a little bit, and let's take a little bit of red here. If we look around this whole muscle belly here, okay, it will be surrounded by, let's see what we'll see there, by the epimysium. So there's the epimysium which surrounds that whole muscle belly itself. Okay. Then if I go a little bit further, and I have the perimysium. So here's a bundle of fibers right here, and that perimysium would be surrounding this whole area right here. Well, a bunch of individual fibers that we find that would be inside there. So if we look again, there's the perimysium around the individual fibers that we have there, and there's the endomysium, which is basically surrounding the individual sarcomere. That's a not really good draw right there. The individual, so around each individual sarcomere would be that endomysium, which would be surrounding it. Okay, so that's a little bit about what we see in regards to the skeletal muscle anatomy. Microscopically, again, that sarcoplasm is basically the fluid-filled environment inside each sarcomere. Okay. When we look at a muscle is actually just a collection of a number of sarcomeres that are lined up in long fibers. Okay, and these fibers then are collected into bundles, and these bundles into larger bundles, into a larger bundle, into a muscle, and stuff like that. And what happens is if I look at an individual sarcomere, okay, we have what are called myofibrils. Now this will be a little bit more explainable when we get to our next video that talks a little about how muscle functions. Inside each individual sarcomere, there are also a subdivision of small fibrils that are inside there, which are essential, which are the reason why, which are the function, which are the way that muscle will contract or shorten. They slide over each other. If you take your fingers and separate your fingers of one hand from the other, and you sort of like intertwine, the tips of the fingers, what will happen is these are the fibers, and these fibers, what they do is if I move one hand towards the other, they will sort of overlap, and as a result, that's what we see how muscle actually contracts and shortens in length. And again, this is just looking at the sarcomere that's been cut across. And you can see right here, all these little dots in there are these little myofibrils, which we'll talk about later on. So you don't have to worry yourself about it too much right now, but we'll talk a lot more about that a little bit later on. Now, this is that later on. So hopefully you got your seatbelts fastened and ready to go from here. And let me show you what we're talking about here. Let me throw up a little picture here. Now this is a sarcomere. Sarcomere would be from this line right here to this line. And this is where the end of that sarcomere would be, and that's called the Z line or a Z disc. It's right here at the end, right here. So this sarcomere or muscle cell goes from Your muscle cell goes from this point here to this point here. This Z disc to this Z disc that's right there. Okay. Now inside there, let me get rid of that. Let me get rid of these. Whoops. Let me get rid of these. Here we go. If we look inside that muscle fiber itself, there are small little fibers. Now if you look here, there's these small little teeny little ones that are right in here, and these are called actin. Actin is a protein myofilament, and basically the end of the actin down here, if you look at where that actin is, comes down this way. If you follow the little pointer, comes down here and attaches right here to the Z disc here. So this actin filament goes from about here to about there, here to about there, here to about there. Same thing on this side, here to about there, here to about there, here to about there, and they're all attached to the Z disc. The next sarcomere would have the more actin attached to the Z disc going the opposite way. Okay, what happens if you look here, there's some darker red stripes here. These darker red stripes are called myosin, and this is another thicker protein. It's in the center of it. Now this is where it's important, right here in the middle. Okay, and let me draw a line right here, hard to, right here. This area right is called the M line. Now the myosin is attached right to the middle and it's sort of like anchored at this point right in the middle of the sarcomere, where what happens is it can't, you know, that we have part of the myosin going this way and myosin going this way. Guess what? It's stuck right there. So when it contracts, we talked about a little bit more about, just to give you a little idea right here, is here's my myosin, the darker red. Here's the actin, which is the lighter, darker. What happens is, just imagine how what happens is this actin and myosin interdigitate more. What's going to happen is it's going to bring one Z disc closer to the other Z disc. So this one's going to be coming in this way, and this one's going to be coming in this way. And what that does, that overall, instead of the sarcomere being this long, now the sarcomere is this long, and that's because of the interdigitation of all these little fibers. Okay, so that myosin is that thick fiber that we talked about, and it's again anchored in the middle at that Z line. Okay, now that the myosin has little paddles along its length, and what they do is it has some cross bridges to actin. Okay, and these paddles in this area also have areas which will be binding sites for ATP. Okay, and ATP is basically an energy source, adenosine triphosphate, which is a very common energy source used in the body. Okay, and that's how we use the energy to be able to cause this sarcomere from going to this length to going to this length. Okay, also I should talk about, besides the actin, those small filaments, and the myosin, the larger filaments anchored at the M line. Remember, the actin anchored at the Z lines on both sides of the sarcomere, myosin anchored at the M line right in the middle. And again, when they interdigitate, contract, and they overlap, it pulls this Z line this way, this Z line this way, and the muscle cell gets shorter. We also have what's called tropomyosin, and tropomyosin is actually bound around. It's a material that's bound around the actin, and what it does, it actually blocks the myosin from actually binding. Okay, something is going to have to happen down the road. We want to talk about that in a little bit, to actually unlock those binding sites. Okay, or unlock the tropomyosin to allow binding between the actin and the myosin to occur. We also have troponin, and troponin is attached to the tropomyosin, and this has, oh, here we go. Here it has calcium. Remember we talked about calcium and bone. Everybody was thinking that calcium, the most important thing about calcium is for strong bones. Calcium is really, really important in muscle contraction, and that's going to be important in a minute as to how we talk about how this occurs. So anyway, that's a little bit of a basic. Now if I look at an electron microscopic view, this is what we actually see. And remember when we saw those striations? The reason why is this is the sarcomere going from a Z line to a Z line, from here to here, here to here, Z line to Z line. So that's one muscle cell right there. But what happens if you look inside, there's a stripe, and there'll be another stripe, and that's what gave that muscle that striated appearance that we saw when we looked at it underneath the microscope. Okay, that these cross striations that we get, where we have, and what they are is basically, we'll see it right here. See it right from here to here. That's darker here. It's it from here to here, and that's the area where the actin and the myosin are overlapping right at that point. Okay. And this is looking at my actin. Okay, here's the myosin right here, and the myosin has these little cross bridges, and these are going to be important. If you have like a line where you like where you're greeting people, you shake somebody's hand. Well, not now. You're gonna be banging elbows or whatever the case may be with all the coronavirus. What happens if you have and you just or like pulling a rope. You grab a rope and then you pull it, then you grab the rope a little further and you pull it, and you grab it a little further and you pull it, grab a little bit fuller and you pull it. Well, the same thing happens with these little paddles. They're along the myosin. The myosin is going to actually want to grab the actin to pull it to actually shorten that distance, to bring it to increase that overlapping between the myosin and the actin. Okay. Well, again, we talked about how that tropomyosin is over the actin and over the actin filament, and what that does is sort of stops it. But the troponins there, which has calcium binding, what happens eventually, the calcium is going to bind to the troponin, which is going to allow the unblocking of the myosin on the actin, and as a result we'll have that like rope pull type thing, or like handshake down a line, pulling things to be able to shorten the distance between one Z line or Z disc and the other Z disc. Okay, and again, when we look at these bands and lines, okay, we also, when we look at the electron microscopic picture, an A band. Okay, the A band is basically the point from where we're talking about the myosin. Here's the myosin, this darker filament right here. So here's the myosin right here. From this point to this point is what we call the A line. When you start to look at it, it's a dark band of myosin with overlapping actin. Here's where the actin right here is overlapping the myosin. Here's where the actin right here is overlapping the myosin. The I band, okay? The I band is this area out in here, which is basically what we'll find is that we're areas of actin only. So therefore it's going to be much lighter. This area is going to be darker, this area is going to be darker, this area is still going to be dark because it's very thick with the myosin, but these areas are going to be light under the microscope simply because it's actin, and actin is a very thin filament. And then right in the middle there'll be a Z disc, and there's the actin going out this way, actin going out, actin going out this way, and then all of a sudden out here we'll have myosin that'll be in here. So from the end of the myosin to the end of the myosin on the next sarcomere is called the I band. The M line is this area right here. That's right in the middle, and that's the center point of that anchor point of attachment of the myosin. It's right in the middle of the sarcomere. Hopefully that's a little bit explainable to you. We'll be answering those questions that you might have. Okay, and again, this is what we're looking at when we start to look at that electron microscopic picture. Here's my M line, and that's the area right here. You can see it where the attachment, the center anchor of the myosin. Here's my A band. My A band is basically where I see my myosin. Okay, because this is my length of my myosin. This is the Z disc and Z disc, so a sarcomere goes from here to here as this shows. Then what happens is this is part of another sarcomere that's attached. This right here would be the end of the A band. Right there would be the end of the A band. So this area right in here is called the I band. It's light simply because it's only actin in there. This is dark because it's mostly myosin. Okay, and these are even darker because it's this area right here, and this area right here is where I have my actin and myosin overlapping each other. There's no E Street Band, unfortunately. I don't know if you're a big Bruce Springsteen fan. Unfortunately, no E Street Band in muscle fibers. That's a little non-humor. Okay. Now, what's important in regards to that muscle also, they're called transverse tubules. Okay, and what happens is the transverse tubule, if I look at it, go back to an image that we had before, here's a sarcomere, here's the end of a sarcomere, here's the end of a sarcomere. So this is the muscle cell right here. That's a sarcomere right there. So this is going to be a part of another sarcomere, part of another sarcomere. But inside, what happens is the sarcolemma has these small invaginations or troughs that are inside there. And what this allows materials such as, there's a good friend, calcium, to come back to be able to leak in, to cause the ability of the actin and the myosin to actually start to cross over each other. Now, I put magnesium in here, and magnesium is also important. And magnesium is like the kissing cousin of calcium. I mean, these two are it's a match made in heaven. I'd write it in here, but basically sometimes if people have a calcium deficit, they could actually be given magnesium, and magnesium will actually help because they're chemically very similar and they do a lot of the same things. So what happens is these invaginations are called T-tubules, actually then leak down through the sides, and get to the ends. You can see it here on the surface. What happens is it's going to go along the whole end of the area of the sarcomere to allow infusion of things such as calcium, magnesium, and stuff like that, which are essential for muscle contraction to occur. Okay. Um, Muscle contracts in response to motor nerve. I mentioned this before. Muscle, we know skeletal muscle is voluntary, which means that I have to tell it with my brain saying, do this, do that, do this, do that. So it has to respond to motor impulses from a certain area of the brain, which we'll call the motor cortex for now, but we'll show you exactly where that is in about another couple weeks, or another week maybe, okay? And what happens is once that portion of my brain in that motor cortex says, muscle, I want you to do this. The impulse goes down, actually goes from the right side, it crosses about most of those fibers will cross from the right side to the left will cross most of it from the left side to the right, and it goes down the spinal cord to eventually exit out through the spinal cord. Remember if we looked at in the lab how those little gaps between the vertebrae allowed spinal nerves to come out. What those spinal nerves are basically a collection of a number of nerve fibers that will go to a certain area on a muscle, okay? So it sends this impulse that starts in the motor cortex down through the spinal cord, out through a specific spinal nerve, and finally then what gets to the end of a muscle into what we call the motor end plate. And this motor end plate is that point where that nerve will finally contact a particular skeletal muscle. When this signal gets down to the end of that nerve, inside the end of the nerve are small little vesicles or small little bubbles that contain what's a neurotransmitter, and this neurotransmitter is called acetylcholine. What happens is when the signal finally gets down to the end of the nerve, it's this trigger to allow this acetylcholine to be released into the gap between the nerve and the sarcolemma, or that muscle cell membrane, okay? This is also called the neuromuscular junction, okay? And so this motor end plate neuromuscular junction is basically where all the magic happens, where we have that communication between a nerve, the nervous system, and between the muscle, okay? And this area, this motor end plate, has a small cleft between the end of the nerve and the sarcolemma, where we have these chemical reactions that will occur that will allow that muscle contraction to finally exist. Now, again, that neurotransmitter is called acetylcholine. It's really an important type of a neurotransmitter. There are lots of neurotransmitters. There's some that are special in the brain, serotonin, a number that we have, but the one that we have in regards to muscle is called acetylcholine. Again, what happens is when that nerve reaches that motor end plate, okay, and that what will happen is it causes the release of that neurotransmitter, the acetylcholine, into what we call the synaptic cleft. That synaptic cleft is that space that we have that's between the nerve and the muscle. And this is what actually causes the muscle to become stimulated. What happens though is that if you think about that, if the acetylcholine is causing contraction, we don't want that acetylcholine to be there forever. So as a result, what happens is there are small little gaps small little clefts within that synaptic cleft, okay, which actually have another enzyme, which is called cholinesterase. Again, ASE, if you see ASE at the end of a word, it's probably an enzyme. And what happens is cholinesterase will actually deactivates the acetylcholine. So therefore, once the acetylcholine works, it can't continue to work to keep the muscle continuously contracted, but it will work, the muscle will contract, and then the cholinesterase will actually deactivated, actually breaks down the acetylcholine, and the acetylcholine gets transported back into the end of the nerve to be able to make more acetylcholine eventually. A couple of things we have to think here again, and some things we've already briefly alluded to, is one motor neuron generally stimulates a group of muscle cells, okay? What I mean is that what happens is when that nerve gets down close to a muscle, we can't have so many nerves that there's an individual nerve for every sarcomere. That'd be just too many nerves. So what happens is that nerve will branch. When it finally gets down to that area close to the muscle, this motor nerve will actually branch off and will supply a number of closely associated in area muscle cells, sarcomeres, and this is called a motor unit. So one motor neuron generally stimulates a group of muscle cells, and that's called a motor unit, okay? Again, a couple things about it is you think about it, sometimes you only have to exert a little bit of force, sometimes you have to exert a lot of force. And this is what happens is forceful contraction is what we call recruitment. What happens is recruitment simply means the more motor units that are stimulated, the stronger the contraction force is going to be. So therefore, this is important. Where what happens is each individual motor unit is what we call an all or none phenomenon. Once that signal gets down to a motor unit, it's going to stimulate that sarcomere. But the more motor units that I activate, okay, by more nerves, more motor nerves, that's what we call recruitment. So therefore, more sarcomeres are contracting because more motor units are being activated. Okay, a couple of things to note about this concept of motor unit and recruitment is that muscles that need fine control have smaller units, motor units. And this makes a little bit of sense. In other words, if I need fine-tuned control, I need to have more nerves going to that area that supply a smaller number of sarcomeres each. Okay, and that just provides a lot more control. It's logical when you start to think about it. Muscles that don't need a lot of fine control are like for maintaining posture and things like that, have larger motor units. In other words, what we'll have is one motor nerve will supply many more sarcomeres, where muscles that need fine control, one motor nerve will actually supply very few sarcomeres just to provide a lot more control. It just gives more neurological control over that muscle contraction. Now, again, I mentioned that area where that motor neuron contacts the muscle is called the myoneural junction at that motor end plate. Myo, muscle, neuro, nerve junction. Okay, and again, that neurotransmitter that we see is called acetylcholine, which stimulates the muscle fiber to contract. Okay, most muscle fibers have only one motor end plate. Now, this is different from motor unit itself. Motor unit sort of relates to the number of sarcomeres that will be supplied by an individual muscle fiber. However, fibers, about 98%, only have one nerve fiber that will stimulate them. In other words, you can't have a sarcomere with two different nerves coming and contacting that individual sarcomere. It just doesn't work. Okay, it's sort of like like a tug of war between two. So most muscle fibers, about 98%, only have one motor unit. And I sort of put this maybe to explain it, where one neuron generally innervates a number of sarcomeres. A single sarcomere generally has only one point of innervation. Okay, and when I say innervation, I'm talking about supply from a nerve. Okay, a couple anatomical considerations to consider. The motor end plate. The motor end plate is the enlargement at the end of the motor nerve. What happens is when the nerve gets down close to the muscle fiber itself, it starts to bulge out, and that's this enlargement that we see at the motor end plate. Now, the axon is basically that long wire or that filament. We saw that a little bit in the lab when we looked at nerve fibers. What happens is we know that what happens in the brain, we have a cell body, and the cell body actually leaves the brain and usually synapses, which synapse means it actually connects with another nerve, and then finally that nerve, which has a very long axon, a very long filament, that will actually come down through the spinal cord and exit out in that little gap from the side of the spinal cord, from the vertebrae, as we talked before, from the spinal cord. Basically, that axon is generally covered with something called myelin. Okay, and this myelin is produced by certain cells, and this myelin is produced by cells called Schwann cells. And what the Schwann cells do, and we'll talk more about how the Schwann cells work, but it actually speeds nerve transmission. So it allows that signal that has to go a long way to go a lot faster. Muscle or nerves that are non-myelinated or unmyelinated are slower. Nerves that are myelinated, because these Schwann cells make this myelin, it's like an insulation that covers it, increases the speed of transmission of that nerve. Okay, what happens is then finally when that end of that nerve gets down to the muscle, the sarcolemma, it invaginates. It creates there's a little bit of a dent of the muscle fiber, but it doesn't. The nerve does not penetrate through. It creates a gap, and that indentation that we see in the sarcolemma is called the synaptic gutter. Okay, and then between the nerve, therefore, there's and the muscle, the sarcolemma, there's a gap, and that's called the synaptic cleft. And that synaptic cleft is filled with gelatinous material and extracellular fluid that actually provides a liquid environment for things such as the acetylcholine to be able to move. It's about 200 to 300 angstroms wide, but I wouldn't expect you to remember that. I'd never ask anything like that on an exam. And this is what we're talking about. Here's that motor coming down. Here's the end portion, the axon terminal, where the end of it is. And it causes an invagination of the sarcolemma that comes down this way. Okay? So this invagination would be called the synaptic gutter. And the gap that we have between the two is called the synaptic cleft. Synaptic cleft. Now, in the end of that axon terminal, again, we had these vesicles, these little bubbles, and these little bubbles are filled with that acetylcholine. We'll talk about that. And then we're talking down here, we're getting down to the sarcolemma. You can see the actin, the myosin, sort of like in there. So, but that's just a little artist rendition. A couple of things I should mention about the axon terminal is there's a lot of mitochondria in the axon. Now, what do the mitochondria do? Mitochondria, we know, are for energy production. We need to produce a lot of energy. Why? Because the axon terminal needs to be able to make a lot of acetylcholine. So we need that energy to be able to provide the production of the acetylcholine that's synthesized in the cytoplasm of the axon terminal to be able to be put in those vesicles to be released. So for this to happen, we need a lot of energy to produce the acetylcholine. Therefore, the mitochondria are increased in number in that area of the axon terminal. And acetylcholine is basically a combination of acetylcholine plus choline, and that process, the acetyl-CoA plus choline, is made into what we call acetylcholine. Okay, and it's enzyme acetylcholine acetyltransferase that catalyzes this response to this reaction that happened. So what happens is, we talked about before how the cholinesterase breaks down the acetylcholine so it doesn't keep on stimulating the muscle. What happens is that the breakdown products get transported back into the axon terminal, and basically with the acetyl-CoA and the choline as breakdown products of the acetylcholine, this enzyme choline acetyltransferase will actually take and combine these two things and produce those into acetylcholine, and that's where we get the acetylcholine. Once the acetylcholine then is made in the axon terminal, they're stored in vesicles in the nerve endings. Okay, and again, these are neurotransmitters. Okay, this is what we talked about before. Here's that axon coming down. Here's the myelin sheath that's around that would speed that conduction. Here's the axon. Here's the terminal right down in here. Vesicles, the mitochondria to make the energy to be able to make the acetylcholine. And then finally what happens is the acetylcholine then gets released into the synaptic gutter. Okay, that's what you need to know right now. But this gets a little bit more complicated in just a second. What happens is once that acetylcholine gets into that synaptic gutter, what happens is there are receptors on that sarcolemma for acetylcholine, and the receptor binds the acetylcholine. When that happens, that starts the ball moving. Everything starts to move from here to cause that muscle contraction to occur. Okay. Now inside that synaptic cleft, there are what are called secondary synaptic cleft, and that secondary synaptic cleft is important for a couple reasons. Number one, it increases the surface area, which means that there's more places for the acetylcholine to bind. Second thing is, within that secondary synaptic cleft is that cholinesterase, and that cholinesterase and these secondary synaptic clefts are what will destroy the acetylcholine and transport them back to the area of the axon terminal, so I can make more acetylcholine from the parts that are broken down by the cholinesterase. Again, ASE enzyme, and the enzyme breaks down the acetylcholine. So the choline is active, is resorbed and reused back in that axon terminal. Okay. The acetyl-CoA comes from another area, but the choline from the acetylcholine gets reused again. Okay. And this is what we're talking. This is motor end plate here. Here's the axon, or the nerve terminal here. This would be the synaptic cleft in this area right here. And then at the corners, the areas here, we have areas that are areas of receptors for the acetylcholine to be able to attach to once they're released from all these little vesicles that are up here in the area of the axon terminal. Okay, so just to summarize what happens in neuromuscular stimulation, what happens, a nerve that starts in the brain eventually reaches this motor end plate. Okay, the nerve impulse reaches that motor end plate. That's point number one. Actually, we'll talk more in the neurological section about what happens before that. Okay, so it reaches the motor end plate. At that point, there's a stimulus to release the acetylcholine from the vesicles. The acetylcholine that's stored in the vesicles now is released into that synaptic cleft. So that nerve action potential causes a shift of calcium from the extracellular fluid into the membrane of the terminal, which causes a rupture of the vesicles and the release of acetylcholine. Whoops, there we go again. There's our good old friend calcium coming back to haunt us again. So calcium is one of the triggers that when finally that signal, the electrical signal, gets down to the area of the terminal end of the nerve, it causes a shift of the calcium from the extracellular fluid in the end of the nerve into the membrane of the terminal, which then allows the acetylcholine to be released into that synaptic gutter or synaptic cleft to be able to stimulate the sarcolemma. Okay, acetylcholine then diffuses, like I mentioned, to the synaptic cleft. At that point, it binds the acetylcholine receptors on the muscle fiber membrane, which we talked about, and that's the stimulus for contraction. Muscle fiber then contracts, and that's how we get a muscle contraction. Well, sort of. There's going to be more about that that's going to come up. Let me just show this again. Here's again that axon right here's the terminal axon. Here's the terminal right there. Here are all the vesicles with the acetylcholine. Here's the synaptic gutter. The cleft is in here, and finally here are my secondary synaptic clefts with the cholinesterase and stuff like that will help to break down the acetylcholine, so part of the choline can be put back into the axon terminal to be remade into more acetylcholine. So acetylcholine from the vesicles enters the cleft. Okay, as we talked about before, acetylcholine binds to the receptors on the muscle fiber, the sarcolemma, and causes a change in the membrane permeability. There's an influx of sodium at that point. Oh, here we go. Remember we talked earlier, and you're probably wondering about what's the importance. You probably hear my dog barking in the background. There's some dog outside, I guess. It's late at night, but somebody's out there walking the dog, whatever the case may be. But anyway, sorry about that. Quiet, Quinn. Anyway, what happens is we talked earlier in the year about sodium and potassium moving across the membrane. What happens is at this point we have a change in this membrane permeability, and that occurs because that sodium rushes in. I can't write it in very well. Sodium rushes in, which causes a change in the electrical status around that membrane, and it's called the membrane potential. Okay, as the sodium rushes in, what rushes out? Hopefully you said it. Potassium. Potassium rushes out to try to balance that, but that's what happens. Okay. What happens is eventually there's enough of an electrical change to create what's called an end plate potential. The end plate potential is a strong enough electrical signal to be able to cause that muscle fiber to contract. Okay, if it's sufficient, if that end plate potential is sufficient, it produces what's called an action potential. And the action potential is basically that electrical activity that now disseminates itself and spreads itself along that whole sarcolemma to be able to stimulate that whole muscle fiber and cause my actin and myosin to do their magic by overlapping each other, and that's what we call a muscle contraction. So electrical stimulation all along the sarcolemma, which then causes the changes in the filaments that they start to interdigitate or overlap each other a lot more, which then causes that sarcomere to get shorter, and that's what we see in a muscle contraction. It's just that simple. Okay, and again, the same thing, we've seen this, so we don't have to talk about that before. Oh, this is good here. What's that? That's my lipid bilayer. Remember we talked about the lipid bilayer? If you don't remember that, go back to the beginning of the year. We talked about how cell is amphipathic, and some are polar, non-polar areas and stuff like that. It actually keeps fluid in, but it has a lipid bilayer. Okay, go back and review that. And again, here we're talking about there's potassium sodium channel. Here's that channel we talked about earlier in the year. So you should be able to go back and review that as well. It takes about 2 to 3 milliseconds for acetylcholine to stimulate a muscle fiber, which is really short, okay? And then within that 2 to 3 milliseconds, what happens is some of the acetylcholine diffuses out of the cleft, so it's lost. The remaining acetylcholine that stays inside that synaptic cleft gets inactivated by cholinesterase, which is sort of like hiding in these little secondary synaptic gutters and these little folds of the gutter, and then it'll actually sort of like break down the acetylcholine, and that choline can be then put back into the area of the axon terminal to make more. And this prevents a re-excitation. In other words, we want the muscle to contract and then stop. And if I want to contract again, I have to keep on sending more muscle signals, or excuse me, more nerve signals down to the muscle for that to occur. So the end plate potential is responsible for that action potential. Again, if that end plate potential gets to a certain level, it creates an action potential, and that's the electrical stimulus that goes across the whole muscle. Now, these are some numbers, and I'm not going to probably hold you to it too much, but the resting membrane potential is about negative 85 millivolts. Okay, and what happens is we need to raise that to about negative 50 millivolts by that change of that sodium rushing in to produce an action potential. And that local end plate potential is between about 50 to about negative 7 millivolts. And basically what happens if I get above this 50 millivolts, then what happens, that's generally sufficient to be able to produce an action potential. What this means is that an electrical charge change occurs which stimulates the membrane again, and that's because of that sodium rushing in that causes the change in the polarity from the outside of the cell to the inside. And that's what, and again, the electrical charge change occurs due to the inrush of sodium from the outside of the sarcolemma, and we talked about that earlier on in the year. And this is just a little drawing of the action potential. Here's the resting potential down in here, and here's an end plate potential. So here's my negative 85, 90 millivolts, and that's what the muscle is at normally when it's resting. What happens by the stimulation that we get with the acetylcholine interacting with the acetylcholine receptors in the surface is eventually that causes that inflow of sodium that causes that change. All of a sudden, electrical potential starts to rise, and when it gets above this area, which is that threshold, all of a sudden, anything above this is significant enough to create an electrical stimulus that now goes down the whole muscle sarcolemma, and that's what we see, and that's what causes that muscle contraction to occur. The action potential is transmitted in both directions along the muscle fiber. So let's say if I have a sarcolemma like here, and that motor end plate is right here, that action potential is transmitted both ways along the muscle fiber itself, and it stimulates the muscle fiber to contract. So in there we have our actin and myosin that are overlapping, and that causes the muscle to eventually contract. Okay, now I'm going to leave this for now, and I'll bring this up in another lecture, various problems. I think that's enough for now. I'll actually bring up this as a second lecture in regards to problems, because there are a number of different problems that we have to talk about, such as in the motor cortex, the spinal cord, the peripheral motor nerve, the neuromuscular junction, and the muscle fiber. And so we'll talk about those a little bit later. And I think what we're going to do is we're probably going to talk about the sliding. I'll wait to talk a little bit about the sliding filament theory of Huxley. Let me just zip right on through these real quick, and I'll pick that up in a little bit later on with another